Sam, for raising everybody's expectations of my talk here today. And I've had many graduate students, and Sam certainly was the most exceptional student I've had. So I'm very fortunate to come here and be with you all, see Sam. I'll be talking today about roots of the second green revolution. And so here we were in the 1960s. If we fertilized plants, we would actually lose yield, plants such as wheat and rice, because they would get tall and fall over, they would lodge. So Norm Borlaug and others developed dwarf varieties of wheat and rice that could not fall over, and therefore you could fertilize them and get an increase in yield, although there was no yield gain of low fertility. That was the basis of the historical green revolution. What I'm going to be considering with you today is the possibility of doing what's on this red line, which is a good yield at high fertility and better yield than we currently have at low fertility. So that's the area of the historical 20th century green revolution, perhaps the most significant agricultural innovation of the last century. And this is the area of potential benefit of the 21st century green revolution. Well, do we need a green revolution, a second green revolution? Currently, 850 million people suffer chronic hunger. That number is growing in Africa. Over 3 million children dying of hunger every year. Half of all childhood deaths in the developing world are related to malnutrition. And of course, the human population is expanding. Virtually all of that expansion is going to occur in food insecure regions. In these regions, low-input agriculture is what is practiced. Agriculture without fertilizers, irrigation, or pesticides. And so this is low-input agriculture. In fact, low-input agriculture is the single greatest human occupation. Low-input agriculture is low-output agriculture. Here's some data on yield from different parts of the earth. The top bar is what you can get under ideal conditions, and then the other bars are what's actually achieved in different parts of the earth. Maize, the most important food grain crop on earth, has 20 tons per hectare potential yield under ideal conditions. In Africa, you're lucky to get a ton. You're losing 95% of your yield to some stress factor. Rice grown in water, you're losing 80% of your yield in Africa. Beans, which fix nitrogen from the air, we're losing 90% of our potential yield. So stress, especially nutrients in water, are a principal limitation to food security and food production, economic development in developing countries. You'll notice even in rich nations such as the USA, we have significant stress. But in places like Africa, it is a severe, pervasive limitation. Drought, of course, is one of the principal limitations. And this is expected to significantly worsen in this century. This particular scenario that was forecast a couple of years ago is alarming, and we would not be able to sustain agriculture as we now know it under these climate scenarios. Nitrogen, another big limitation. This map shows you in a color scheme the nitrogen balance in different parts of the planet, green being a surplus from one year to the next, and you can see China, for example, is over-fertilizing. These brown colors represent a deficit, meaning you have less nitrogen each year than you had the year before. Phosphorus is another important limitation. Any red color here represents suboptimal phosphorus availability. So this is a pervasive limitation to productivity when we don't have fertilizers. And the soil base itself is degrading. This is a core scale map from the United Nations showing you soil degradation, largely erosion. You can see even rich countries like ours have serious erosion problems. In Africa, it's very severe. It's estimated recently that 75% of the cropland in Africa is degraded. And this is worsening every year. So what can be done about this? Integrated soil fertility management consists of fertilization when you have access to it. Come back to that in a minute. Soil management, which is very important, although not really practiced in most of the world, including in the US. My neighbors are farmers. They don't practice good soil conservation. This is the richest country on earth. In Africa, people may not know what to do or may not have the resources to do it. So this is very important, but actually it's not very effective. And finally, growing plants that are adapted to stressful environments, which is what I'll be talking about today. Before we go on to that topic, let me come back to this because I think in rich countries like ours, we think, why don't people just fertilize? This is a recent map from the World Bank showing you world fertilizer use on a log scale. You can see these orange and red colors are single digits of NPK per hectare of arable land. These are totally inadequate. And in fact, you see in Africa here the negligible levels. And in fact, these levels are dropping from year to year. One of the countries we worked in, and we work quite a bit in, is Mozambique. And in the past 10 years, fertilizer use has dropped 90% in Mozambique. because of the cost of fertilizers going up because of energy costs. Fertilizer in Malawi costs 10 times what it costs in Utah. So the strategy of fertilization, apart from any sustainability concerns we might have, is just not going to happen in the foreseeable future because of economic limitations. Which brings us back to getting the right plants. That's what we're talking about today. So the first story I'm going to tell you is about this plant, common bean, Phaseolus vulgaris, most important food legume on Earth, an important source of nutrients for about 1 billion people. Typical bean production system here in the Andes. Here's somebody's home. And next to that home, you'll see this very ancient maize bean cropping system. Now this is a low phosphorus landscape. It's volcanic soil. It's been eroded. It's very low phosphorus. If we look at the plants close up, we'll see symptoms of phosphorus deficiency. I would expect this bean plant to lose 90% or so of its yield due to phosphorus stress. And in fact, as I just showed you, average world yields are only about 10% of yield potential. So I started working on this in 1987. I got my PhD and went to work at the International Center for Tropical Agriculture with headquarters in Colombia, South America, where for 20 years they've been screening hundreds and hundreds, thousands of bean lines trying to find anything that would be better than this Brazilian landrace called carioca. And after 20 years, 4 cycles a year, tens of thousands of lines, they didn't find anything that could beat carioca. They fired their soil scientist a few months before I arrived, and the breeders wrote a paper saying it was impossible to beat carioca. I wasn't really happy with this conclusion. I didn't think this was the way to do this kind of work. When you look at yield in the field at one location, you're looking at the effect of a lot of different environmental factors. So for example, this plant here is suffering from some kind of disease. That's perhaps why it's not yielding well, apart from any phosphorus issues. Furthermore, this is the final stages of their germplasm screening. They were intelligently selecting for high yield potential and disease resistance, and then just throwing all that stuff that they had selected into this purgatory plot to see what could survive phosphorus stress. So they'd already narrowed their germplasm base. I thought what we should be doing is what people do for breeding for diseases or insect resistance, which is you find a useful resistance trait and you put it in the right genetic background to make a new plant. I thought that was scientific breeding, and that's what we should do here. To do that, we had to know what the mechanisms were of adaptation to low phosphorus. To make a long story short, we found out what some of those mechanisms were. And basically it involves putting the roots in the topsoil where the phosphorus is. Phosphorus is immobile, does not move in soil, tends to concentrate in the topsoil. So root architectures that concentrate foraging in the topsoil will do better than those that don't. And here's some traits involved in that. I'm not going to go through all these. I'm just going to show you a couple of examples. This is one we found early on, which is the growth angle of the most important root class in bean, which is the basal root. You can see in this case, these are plants dug up in the field. These are shallow, and these are deep. And so this will be doing better at topsoil exploration. To convince breeders of the utility of this trait, we make a genetic cross between parents such as these and get a set of recombinant inbred lines that have the same genetic background but vary for their phenotype of interest. So in this case, these are all recombinant inbred lines from the same 2 parents. And here's the root shallowness in the field, and here's their phosphorus uptake, and this is about a 6-fold variation in phosphorus uptake associated with variation for this root trait. With these RILs, recombinant inbred lines, we can also do genetic mapping. So here's the 11 chromosomes of common bean, and we have segments of the chromosome that are associated with performance in low phosphorus fields in the tropics. Those are shown in red, like you can see here, some here on chromosome 11. And then the yellow, blue, and green are regions associated with root shallowness that we measure in a plastic bag in the laboratory. And you can see here regions such as, once again, on chromosome 11, where we have co-segregation. We have genes involved with both root shallowness and performance in the field and under low phosphorus. That means if breeders select for root shallowness, they're also going to get a benefit for yield in the field. Here's another trait. This was something that was known before we started researching this topic, which is root hair length and density. Root hairs are subcellular protrusions which extend out from the roots into the soil and expand the zone of soil that can be used for phosphorus acquisition. This is the result of scientific breeding here, and this is a landrace from Highland, Peru that's never been touched by scientists. You can see the large variation in root hair length and density. So we take these 2 parents, cross them, get a set of progeny that share the same genetic background but vary for this trait, drove them in a low phosphorus field, in this case in Costa Rica, and you can see a very nice correlation between root hair length and shoot phosphorus content. About 250% variation in shoot phosphorus content associated with variation in this trait. This is a relatively simple genetic trait, only 6 markers, explain over half the phenotypic variation. Now, breeders have to put together phenotypes composed of many traits, an integrated phenotype. And so we were interested in how these traits interact with each other. So here's a study done by Magallanes Miguel in Mozambique, where we had genetic variation in the same set of plants for both of the traits I just introduced to you, root hairs and also root shallow. So for example, we have short root hairs and deep roots. This should be the worst for phosphorus uptake. And so we're considering that our baseline. This is a field study. Here we have a set of plants that have shallow roots but short root hairs. Here's plants that have long root hairs but deep roots, and here's the case where we have both positive traits. So if we compare to the baseline, if we have shallow roots, we get a 60% increase in growth in the field in Mozambique. The root hairs give us a 90% increase in growth. So if we have both traits together, we should have a 150% increase in growth. But actually we observed a 300% increase in growth. That's because those hairs are more useful if they're on shallow roots than they are if they're on deep roots. And I think this kind of phene synergism is very common in plant traits, especially below ground traits. So the utility of a trait is very much affected by other aspects of the phenotype. Another thing we have to be concerned about is trade-offs. A trait might be good for one thing but bad for something else. If we're talking about shallow roots for phosphorus, we also have to be concerned about needing deep roots for drought. That was shown in this study by Melissa Ho some years ago. This is one of Sam's cohorts in graduate school. This is a study in Honduras. We had both drought and low soil fertility. So the same genetic background, we had deep and shallow genotypes. Under low phosphorus, the shallow genotypes did better, as we expect. And under drought, the deep genotypes did better, as we expect. So this is a conundrum. Breeders do not want to produce a line that's good for drought, bad for phosphorus, or vice versa. They want both, because small farmers have to deal with both constraints. So fortunately, in this plant, we were able to discover a trait that will give us both shallow and deep exploration. It turns out in this species that the nodal roots, the basal roots, are produced in distinct positions, which we're calling whorls. You see, this genotype only has 1 whorl, and this has 3. Each whorl can make 4 roots. The more whorls you have, the more roots you have. Our hope was that if we go, for example, from 2 to 3 whorls, we have more positions with which to grow roots, then we can explore both shallow and deep soil with the same genotype. This is what the phenotypes look like in computer modeling. 1 whorl on the left, 2, 3, and 4 whorls here on the right. So once again, this was field work done in Mozambique. You can see the red weather tropical soil in the background. And we have the same genetic background, plants that have 2 whorls and 3 whorls under low phosphorus. We have a 60% increase in growth at low P by increasing the number of whorls, which was good. This is work of Katie Barlow looking at this under drought at our site in South Africa. She was looking at a population that had either 1 or 2 or 3 whorls. If they're well watered, they grow the same. If they're under water stress, you have a 70% increase in growth by increasing the whorl number. This particular trait is under simple genetic control. 2 genetic loci explain over half the variation. So this is a nice trait. In this species, we have a trait that allows it to have both shallow and deep roots. Another possibility is, rather than trying to make one plant do everything, to use a combination of plants that work well together. For example, we could have shallow-rooted plants that are good for low phosphorus, deep-rooted plants that are good for drought, and have them in a community together, which minimizes root competition and also makes sure that some plants will be doing well in your field no matter what the environmental condition. Here's the idea. These are called multi-lines. This is part of the PhD work of Amelia Henry. She's now a rice physiologist at Erie. So we have, these are closely related lines. They look the same to the farmer, but these are actually shallow-rooted, these are deep-rooted. Mixed together, the farmer may consider that to be one variety, but we know it's actually 2 different genotypes that have complementary architecture. So Amelia looked at these in a range of stressful environments, on-farm conditions in Honduras, going from very low yield to better yield. And if we're looking at the multi-line yields in red versus the average of the single-line yields in blue, there was consistently a statistically significant benefit to the multi-line. So here's another strategy. Rather than trying to have one plant that will do everything, we can get a combination of plants that are more stable in field conditions. Another consideration our stakeholders, our sponsors had was that we were going to somehow mine the soil and degrade soil fertility over time. By getting a plant that extracted more phosphorus, we're going to remove phosphorus from the soil. And these are people who don't fertilize, so eventually we might end up degrading soil fertility. That was an important issue, had not been researched. We didn't think it was going to be a problem because of this sort of thinking that we show on this slide. This is based on literature data. We thought, okay, if we have 1 ton of yield, which is twice the world average, there's 2 kilograms of phosphorus in that ton of seed. So every year you're taking out 2 kilograms of phosphorus from the seed. If we double the yield to 2 tons, now we're taking out 4 kilograms of phosphorus. So we're mining the soil. Low input agriculture is basically soil mining. However, the biggest loss of phosphorus in this system is erosion, which can lose significant amounts of phosphorus. By having bigger plants, we're protecting the soil and thereby reducing this flux of phosphorus. Therefore, the net effect is you're actually conserving phosphorus with the extractive genotypes. Not to mention, there's more food and more money and perhaps more fertilizer being used. But even just in terms of soil fertility, we thought the more extractive genotypes would be better. Once again, this is work of Amelia Henry. Here she is, working with Costa Rican farmers. This is a Costa Rican bean field. She rented from these people. She went out and lived with them for 1.5 years to do this study over a long term. She built these lysimeters so she could catch the phosphorus running off the field and measure the phosphorus in the seed. You can see here the existing deep-rooted lines are not covering the soil as well as these new shallow-rooted lines. She was able to show that these new lines not only yield more, but they reduce phosphorus loss by 50 to 70%. So what happened to Carioca, the unbeatable line? Now I cannot convince the bean breeders to even use it as a bad check because we have lines that are so much better. Here's Steve Beebe, head of the CF bean program, some of his data. Here's Carioca under phosphorus stress, giving you 700 kilos per hectare, which is what Carioca normally gives you. This is a very realistic level of stress. These 3 of these new lines with better root traits. This one in particular, look, it's giving us over 2 tons of yield without fertilizer, 3 times that of Carioca. These are twice that of Carioca. So these now are being used as parents. These and other lines we've identified are being used as parents in CF breeding programs and being deployed through Africa and Latin America. Now we have sponsors who are helping us push this technology forward in Africa in a targeted way. We've been working with Celestina Joshua, former student who's the bean breeder of Mozambique, by selecting for root hairs, basal root whorl number, and basal root growth angle. Celestina has released, now last year she released new bean lines that are yielding 2 to 3 times more than the best yielding lines we had previously. This is her husband, Suarez Serinda, is an agronomist, was also a Penn State PhD student. Here's Suarez doing an erosion study. And he's showing that these new lines have much greater nitrogen fixation than the old lines. They protect the soil from erosion. They do not bother the maize they're often grown with, and they're better able to utilize locally available rock phosphate. So we thought this was a great success. We had much more food production and ecological benefits, but our sponsors were not impressed because they don't really care about that. They care about people. We have to show that this new seed types are actually benefiting communities and households. So we've been working with social scientists, especially Jill Findice, who's now at the University of Missouri, to try to figure this out. And so we've done studies for some years, both before the release of new lines and now since the release of the new lines. And we found out some interesting things. This is Carioca, and if you interview lots of farmers, and women are being interviewed by women, men are being interviewed by men when this other gender's not present and so on, so you get the right answer. So Carioca, if we increase the yield of Carioca, Carioca has no market value. It's not sold in the marketplace in Mozambique. It's grown by women to feed their children, which is wonderful because 70% of the families in Mozambique are food insecure. So if we increase the yield of Carioca, the women would have more beans for their kids. That's the outcome we were hoping to achieve. If we improve the yield of this type, this is the main commercial type in Mozambique that is then, commerce is handled by men. And what will men do with the extra income? They will buy beer. This is not the result of just our study. There are many studies in Africa showing that one of the most common things you do with extra income in Africa is buy beer. Now the men will tell you that this actually benefits the family because they do not own their own land. And by drinking beer with the village headman, that they will have a better allocation of land in the next year. At least that's what he says. These particular types of seeds are worth, last I saw in Mozambique, they're worth $1.90 a pound. So you're probably paying something like $1 a pound for dry beans in Utah. These people are paying twice as much as you are. The average wage in Mozambique is 70 cents a day. So this is a very high value crop. If you have access to an urban market and can sell these beans, you can make a lot of money. What do men do with this much money? They already have enough money for beer. They're going to marry another woman. This is a polygamous culture. And so the more successful farmers have more wives. So there's all kinds of interesting social impacts of improving bean yield depending upon the types of beans that you're improving. Another issue that Jill has dealt with is the dissemination of these new seeds. You do not order seeds in the mail there. You get seeds from your friends. That's where you get bean seeds. And so Jill has gone in and done very beautiful studies of mapping the locations of every household and whole villages, surveying all those people, understanding how they share technologies and seeds. And this is just a very, very small piece of that type of network analysis. What she sees then, and among these, there's 4 families in this network. The husband's network tends to be more extensive than the women's network because the women tend to be focused more on the household because they're taking care of children and they're taking care of the beans, whereas the men are trading and drinking beer and so on. So that means that if we introduce a new seed that the men like, that seed is going to go quickly through the community and be disseminated. If we introduce a new seed that the women prefer, that may not go anywhere. And so we've had to devise kind of dissemination strategies that take into account these local social characteristics. But the work is succeeding and we're having significant impact in Mozambique. I'm just bringing up some of these social issues to show you the kind of things that we have to deal with in Africa. This work with bean was seen by the McKnight Foundation as something we could apply to soybean in China, which is a food security crop. This is work done by, in collaboration with Xiaolong Yan, my dear friend and former student who tragically passed away from cancer a few years ago. Xiaolong's team were trying to take this, which is commercial soybean after several thousand years of domestication and growth in China, and then this was our idiotype. This was our ideal plant in our minds that we want for low phosphorus soils. Xiaolong's team was very successful. They've now released 15 phosphorus-efficient soybean lines in South China in provinces inhabited by 600 million people. You can see significant yield gains. This is the best current cultivar in this low phosphorus soil. This is one of these new lines. So they've been very successful through phenotypic selection for these root architectural traits. We've also shown that these same kind of traits and same kind of genetic patterns hold true in maize. And of course, now maize is a very big plant, as you know. It needs a lot of water and nitrogen. So we were interested to see how we might be able to use root traits to improve the capture of these resources in maize. In poor nations, water and nitrogen strongly limit yield and food security. In rich nations, drought is the biggest risk of maize production. Nitrogen fertilizer in the U.S. is the single largest economic, environmental, and energy cost of growing maize. And the cost of fertilizer has risen dramatically over the past 10 years or so because of energy costs. Amazingly, over half of the nitrogen we apply to maize or to any crop in the U.S. is not even taken up. It directly goes into the environment and causes pollution. So we saw this as low-hanging fruit. If we could improve that, every 1% increase in nitrogen recovery in global agriculture is worth $1 billion a year. So this is something we started to think about. How can we get plants to take up better nitrogen? This is what happens in Pennsylvania in the spring. That pulse of red there is nitrate. In the spring rains, that nitrogen from fertilizers leaching down into the soil, being pushed by rainfall. Here's a corn plant growing. And as the nitrate leaches, it goes below the rooting zone and the plants can't get it anymore. So it's kind of a race of catching that resource before it vanishes through the bottom of the system. So the idiotype, the ideal phenotype that we thought of for nitrogen and water capture is called steep, cheap, and deep. Steep being architectural traits, cheap being metabolic traits, I'll describe those in a minute, to get you very deep roots very quickly to catch that stuff before you lose it. I'm not going to go through all those things. I'm just going to show you a few examples. Here's one in the steep category, optimal number of nodal roots. So what are we talking about here? Here's a maize plant. These are nodal roots, crown roots. And you can see this one has relatively few. This one has many. If we look at 5,000 maize genotypes, we see variation from between 5 and in this case 35 nodal roots. This is what those phenotypes look like. So these first ones that come out are seminal roots. Then the nodal roots are here. So this is the low crown root phenotype, and this is like 6, and this is like 36. And you can see that in this case we have so many roots that are so close together, the plant is wasting energy. You don't need that many roots to acquire water and nitrogen because water and nitrogen can move around in the soil. So we thought this was overkill, and the plant was wasting energy, and we really wanted something like this. That was the idea. To show if that was true, we did field work, in this case under low nitrogen. This is work of Petampong Sengulai. Here's our field site in South Africa showing our high and low nitrogen fields in the USA and in South Africa and in greenhouse mesocosms. The low crown root number lines have deeper rooting, better capture of N15 labeled nitrate placed deep in the soil, better nitrogen accumulation, leaf and canopy photosynthesis, better growth, and 180% more yield. So here's the yield data. Here we're looking as we get less crown roots, we have better yield under nitrogen stress. We recently have got data for the same trait under drought. This is work of Yingjie Gao. And so here we're using rainhouse shelters in Pennsylvania. This would be easy to do here in Utah, but in Pennsylvania we need to stop the rain or else we don't have drought. So under water stress in the field, in the greenhouse mesocosms and in rainhouse shelters, the low crown root number lines had much deeper rooting, greater relative water content, better growth, and between 60 and 116% more yield. Here's crown root number, here's shoot biomass, and here's relative yield. So you can see a very significant, very good correlations too, of increasing yield and growth under drought by selecting for plants with fewer roots. So there's an example of one trait of interest. Here's another one, few long laterals. Once again, I think if you asked your grandparents, do we want plants with big, robust root systems under drought, they'd probably say yes. What we're seeing here is no. We want plants that are very economical in the roots that they build because they're wasting energy on those roots if they don't need them. Here are the phenotypes we're talking about, what we call few long on the left, many short on the right. Same amount of energy going into root growth, but being divided up among many laterals here or few laterals here. Significant genetic variation for this trait. Here's what these phenotypes look like in silico. These models are based upon actual measurements of maize root plants. And you can see here, this is fewer, longer roots, and here the more roots sharing that carbon, so they're not growing as long. Once again, field studies. This is under low nitrogen, work in South Africa and the USA and greenhouse mesocosms, showing we have significantly reduced respiration when we have these few long phenotype, deeper rooting, better end capture, better photosynthesis, better growth, and 32% more yield. This shows in the three environments under low N, the few long phenotype is doing better than the many short phenotype. Jen has recently shown this is true for drought also. Here's one of our drought trials. You can see the differences between water stress and well water. This is at our site in Arizona. So in studies in Arizona, Pennsylvania, and greenhouse mesocosms, the few long lines had less respiration, deeper rooting, deeper water capture, which we can see from O18 analysis, better leaf water content, better stromal conductance, better CO2 assimilation, better growth, and 61% more yield. So here's shoot dry weight and yield as a function of lateral branching density. Once again, perhaps contrary to expectations, the plants with fewer roots are doing significantly better under water stress. What do I mean by cheap? Cheap are traits that have to do with the cost of building a root system. So here's one that we thought was interesting. RCA, root cortical parenchyma. This is a section of a maize root showing you the green areas are air spaces called a parenchyma in that root that's often stimulated by drought, by some kind of stress. And we showed some years ago that as we have more parenchyma, more airspace in the root, the cost of that root goes down because it doesn't take any energy to sustain air, right, as opposed to living cells. There's natural genetic variation for this trait. So these are unstressed plants closely related to each other. They're recombinant inbred lines from the same two parents. This one doesn't make any parenchyma. This one's making a lot of parenchyma. So we, once again, went to our modeling approach to see if this trait could have an impact on overall plant growth under stress by having these small savings in root respiration. This is a map of a parenchyma formation in a maize root system based on empirical measurements. It's developmentally regulated, so we know what happens in older roots, we know how fast it happens, we know how it affects respiration, nutrient uptake, and so on. And so we're able to simulate then, in this case, a plant growing in low phosphorus soil without RCA, or with RCA. Everything else is identical. That's the nice thing about simulations. You can keep everything else identical. And you can see over time, the plant with the parenchyma ends up growing more. Because every little unit of root is taking a little less energy to build. It grows a little bit more, takes up more phosphorus. There's more photosynthesis, which is also modeled but not shown here. And so over time, you have this autocatalytic benefit of a small difference in root cost translating into a big difference in growth. So the model said we could actually get significant benefits from this trait. So this is, as stress increases, the increase in dry weight from parenchyma increases up to about 60% for nitrogen stress, phosphorus stress, maybe even more for potassium stress. I have to admit, when I saw these data, I didn't believe them. It seemed like a pretty big effect. Even under high input conditions, we could reduce the fertilizer needed to obtain maximum yield. This amount of nitrogen is worth about $400 million a year to U.S. maize production. So to show if this was true, this is once again Pethempong Sanguilai's work at our field site in South Africa, also in Pennsylvania, in the greenhouse mesocosms. He showed that maize lines with parenchyma had less respiration, deeper rooting, greater leaf nitrogen content, greater CO2 assimilation, better growth, and 58% more yield. So here you can see the yield benefit from parenchyma formation under low nitrogen. This is data on drought, published a couple years ago by Jin Ming Zhu. Once again, using rainout shelters and greenhouse mesocosms. Once again, parenchyma reduces respiration, much deeper rooting, better water content, better growth, 800% more yield. These are significant yield changes I'm talking about. In breeding programs, people are happy to get 2 or 3 or 4% yield increase from one year to the next. If you go in there and say you have an 800% increase in yield, that's very significant. Here's what they look like: water stressed without parenchyma looking pretty bad, with parenchyma looking pretty good. So Joseph Chimungu recently completed a study with us on farm conditions in Malawi, looking at the lines that people were already growing, seeing if they had parenchyma or not, and seeing how it affected their drought tolerance. So looking at 14 farmers' fields in each of 2 regions in Malawi. This is a high parenchyma under drought, low. You can see a significant gain here. Here's high and low. In this case, over twice as much yield in our high parenchyma lines. These are 2 on-station studies in Malawi at field stations, 20 lines at 2 field stations. High parenchyma lines about twice as much yield. In this case, over twice as much yield under drought as the low parenchyma lines. So more than doubling the yield on farming conditions in Malawi. That was very significant. We were happy to see that. Once we saw the value of parenchyma, we thought, well, let's look at some other traits that could make roots cheaper. Here's 2 new ones that people hadn't thought about. One is we could select from lines that had fewer cell files in the cortex, like this, rather than this, or that had larger cells. We'll talk about these next. So this is work of Joseph Chimungu. So here's the genetic variation for the cortical cell file number. This one has more cells, and so it's going to cost more. There's large variation for this trait in maize. And we showed in field trials in Africa and the US, that once again, the same. I'm going through this quickly because it's the same story. Whenever we reduce root respiration, we get deeper roots, and we get better water capture, and we get better growth and performance. And in this case, here's the yield data in Pennsylvania and in Malawi. The red bars are the yield under drought stress, so the reduced cell file number had significant gain, in this case over double. Here a significant gain, in this case over double. So very significant impact of this trait on drought tolerance. Here's another trait that people haven't thought about: cell size. The idea is that larger cells are mostly vacuole. Vacuole is cheap. It's mostly just water, whereas cytoplasm has nitrogen, phosphorus, mitochondria, and so on. So the idea was by going to large cells, we end up with a cheaper plant. Once again, large genetic variation for this trait in maize, tenfold variation in cell size. And it's related to the same kind of things I've shown you before. Less root respiration, deeper rooting, better water capture, better water content, and so on, better growth. Here's the yield data in Pennsylvania, in Malawi. Here it's about a doubling, and here's more than double our yield under drought by selecting for large cell size. So there's been a lot of research over the years on what they call water use efficiency, which is like how much water it takes to fix carbon from the atmosphere through stomates. And I think we might have been looking at the wrong end of the dog in a certain sense, because everything I'm talking about with you today has to do with this part of the equation, which is how much carbon we're spending to get water out of the soil. All the traits I'm mentioning today have to do with this aspect, and they seem to be very, very important in performance under drought. So these lines are being deployed in Malawi through our colleague Dr. Moses Muliro of Luana, and he's having very good success. Well, one of the things you have to be concerned about in working with breeders, they're very hard-headed, pragmatic people. It's all well and good to talk about all kinds of cool root traits, but how are we going to measure root traits on many lines, which is what breeders have to do, go through a lot of genotypes very quickly to find the good ones. So phenotyping is a challenge. So we've come up with the shovelomics technique, which consists of a shovel, or a pediturbiter in scientific terminology, and eager graduate students who just dig up plants and look at them. And it takes about 2 minutes to plot to do this once you get going. And so we're looking at about 25,000 plots per year currently of different species. And so here's an example. This is Eric Nord doing this work in South Africa. So here we've dug up a bunch of maize plants and rinsed them, and then we're scoring them and we're photographing them. From that photographic image, we have software called DIRT, Digital Imaging of Root Traits, to extract architectural parameters. So somebody in Africa could take a picture with their cell phone of these roots, and then when they get to an internet cafe, upload it, and our colleagues at Georgia Tech have this software, will do all this analysis and give it back to them at no cost. Here's an example. So here's a steep or shallow-rooted maize lines. This is work of Hannah Schneider. So she's using 4 years of field data with this very rough and dirty shovelomics DIRT pipeline, and she's doing genome-wide association study where she has candidate genes that are expressed in the right region of the root. This is for root angle, and this is a candidate gene that has something to do with hormone metabolism, so it seems to be a reasonable way to do genetic studies, even though it's a very quick and dirty pipeline. For our anatomical traits, which we used to be doing with razor blades, we developed a new technique called laser ablation tomography. Here's Bob Snyder sitting with the laser setup. This is we got it for a 90% discount from a military lab that was 3 months old. That's your tax dollars at work. So here's a maize root in the last minutes of its life. It's about to be, this is a real-time image of that root as it's being ablated by the laser, and we're photographing it. And then from that image, that three-dimensional image of the root that has been vaporized, we can extract anatomical features such as a parenchyma, which you'll see here in yellow, and we can quantify that. This image took about 30 seconds to generate on this laser system. The whole laser system costs us $25,000. It's run by undergraduates, so it's a very kind of low-tech, low-budget way to do this rapid phenotyping. And we can even look at roots in soil. This is just a handful of soil, did nothing to it, put it on the laser, zapped it. We can see mycorrhizas and all kinds of cool features. So I think there's going to be a nice technique for this high-throughput phenotyping that we need to do. Once we see those images, then we have software to extract anatomical features called root scan. Once again, so this shows you that even from this kind of quick and dirty analysis, we can get genetic signals. This is a candidate gene for parenchyma that's expressed in the root cortex and seems to be involved in programmed cell death. So we can find genes using these kinds of phenotyping pipelines. A broader concern is that, I've shown you this picture, this is a parenchyma in maize roots, is that these architectural traits interact with anatomical traits. So this is a structural functional model of root anatomy called root slice. So here's the parenchyma that we're going to go through here. And so these traits interact with each other in terms of their effect on the plant phenotype. So here, for example, we see the two of the traits I've talked about today. This is a parenchyma, and this is a nodal root number in maize, and this is under low nitrogen, how those interact to affect performance, and if we increase nitrogen, the shape of that function changes. The same for phosphorus. And what that means is the value of a trait depends upon the value of other traits, and it depends upon the environment. Well, in my little lab group, we're looking at over 24 traits, and we're interested in water and nitrogen and phosphorus. There's a lot of potential combinations. In fact, they're factorial. So if you do the math, we actually have about 10 to the 23rd scenarios of interest. And that's a big number. And so computer modeling is one of the only ways you can really explore that kind of decision space. So this is work of Harini Rangarajan, who's took about 1 million hours of computer time to search through all of these possibilities with the genetic algorithm to find the 250,000 best maize root phenotypes, which are shown on this figure for different nitrogen and phosphorus phenotypes. Now, I don't want to jump into the details on this very complex study, but I do want to make the point that I think in biology we hear a lot about the genome and all the fun and complex things that happen in the genome here. The phenome is also very complex. Okay, this is non-trivial complexity we have to deal with at this organismic scale. And of course, there's trade-offs. In engineering, they have the iron triangle, fast, cheap, and good. You can't have all three, you can only have two. Well, take Renkema, for example. I mean, can we have fast and good effective soil exploration that's also metabolically cheap? We've got to have some trade-offs to these traits. And in fact, in case of a Renkema, we know that it reduces water transport across the root because it turns living cells into air. We've shown that it reduces nutrient transport across the root. We're doing research to see if it affects mycorrhizal colonization, the spread of fungal pathogens, penetration of hard soils, rises from microbial communities. All these things could be affected by Renkema. And before we recommend to some breeder in Africa start using this, it would be good if we understood that a little bit more. But there's not very much literature about this. So I think this is a challenge for the next generation of plant biologists to begin to think more carefully about the phenome and all these issues about, well, what is good at the phenome scale, and not just what's the molecular control of these things. Because what we're interested in at the end of the day, what I'm interested in is impact out here, and a lot of scientists begin and sometimes even end over here at the genome scale, and I think we really have to look carefully at the phenome scale to make sense out of this biology. And I think to do that, we have to start developing better conceptual tools. What is a phene? Phene is to phenotype as gene is to genotype. You people may not have heard the word phene before. It was first coined in 1925. Gene is from 1909. We have a lot of words to describe the genome. We have very few words to describe the phenome. In fact, people don't even use the one word we have, which is called the phene. So, instead of thinking of yield as a phenotype, yield is then determined by other processes and functions that are then controlled by individual elements of the phenotype that are called phenes, and we really need a better handle on this part of the equation to make progress with some of these agricultural issues, I think. So to summarize, root architecture is an important component of soil resource acquisition. Topsoil foraging enhances phosphorus acquisition. Subsoil foraging enhances nitrogen and water acquisition. Several anatomical and architectural phenes affect the metabolic cost of soil exploration, which is really important. Bean and soybean genotypes incorporating these root traits are being deployed in Africa, Asia, and Latin America. The steep, cheap, and deep hypothesis or idiotype for nitrogen and water seems to be very promising. We're starting to deploy it in Africa. We'll see how it works. We have high throughput phenotyping platforms and numerous genetic leads. Let's go back to the idea of the second green revolution. Breeding for yield at low fertility has been successful in the few cases it has been attempted. Aluminum tolerance is working and being deployed today. Phosphorus is working. I gave you an example of that today. Nitrogen is more complex, but it should be feasible. Manganese tolerance is being used in breeding programs. Recent advances in root biology are making it possible to design root systems for particular production environments. The bottleneck, in my view, is not more genomic information. It is understanding the phenotypes and phenomes. And we need information about how we get these new phenotypes are going to affect the environment. But I think it's going to be benign. We're not going to lose resistance to drought as we might with the disease resistance. They're not scale positive, meaning they don't just benefit rich people. They would be useful here as well as in Africa. And I think one of the drivers behind this in rich nations, research in rich nations, is we can reduce pollution loads from things like nitrogen and phosphorus. Certainly, as soils degrade, fertilizers become costlier and the world population expands, the need for this is only going to become greater in years ahead. Now, I've been talking about the work of many people. This is the current lab configuration. Thank you very much.
Questions?
So it sounds like these are all, you've been doing breeding studies to get the different phenotypes. Do you do any genetic modification?
No, because most African countries do not accept GMOs. And furthermore, there's no single gene that affects the things we're interested in that is useful. These traits are under complex genetic control, many genes having a relatively small effect in most cases, so inserting some foreign gene just doesn't make sense for this kind of stuff. This is all just natural hybridization.
I appreciate you mentioning that genomics needs at the end and nutrient content of these foods. What kind of studies have you done to show how that changes? You're going through so many iterations of different plant types
Well, actually quite a bit. I mean, the Gates Foundation put several $100 million into what they call the Harvest Plus program, which is all about increasing iron, zinc, and carotene content of beans and maize and rice and so on. And so, and these breeders, of course, they've got multiple traits they're trying to build into their pipeline. So they're working with me to build in the root traits, and they're working with other folks to bring in the iron and zinc, and they've got disease resistance. So they're managing all these different traits to bring together every year to come out with something a little bit better. So nutrition is definitely part of the, human nutrition is part of the equation. But beans, in the case of beans, are very nutritious already. They have a lot of iron and zinc. So, fiber.
Yeah, it seems that looking at a single species, or the individual species traits is important, but could you speak to the role of mycorrhizae and how they might modify this?
Yes. Yes, we have done studies with mycorrhizae, and we've determined they're not important for our work, and so we ignored them. I mean, that seems like a bold statement, but they're very cool organisms, and they're very important, of course. But what I mean to say is when working in breeding and looking at genetic variation, we see that plants are mycorrhizal or not, it doesn't change their ranking. They all get better, they all get worse to about the same extent, and we've looked at that particular issue. So I've shown you a lot of field studies where the plants are mycorrhizal. I've also shown you music cusp studies where they're non-mycorrhizal. It does not change our results. So they're kind of part of the background, and we don't focus on them, which is good because they're extremely complicated to work with. So they're there, and we don't think about them in breeding.
Are the big international seed companies doing anything with this concept of beans? It seems to me that they're not. But
No, beans is not, there's no money in beans. Maize, they're interested in.
No, you missed phenes.
Oh, phenes. I think it's beans.
Are they phenotyping like the way you're describing?
In general, well, the European, we're working with the KWS, which is a European-based seed company, and they don't have GMOs in Europe, and they're interested in phenotypes. The US seed companies are all captivated by genomic selection and the possibilities of genomics, and they're really not interested in phenotypes, and I think they're making a mistake.
So just to further that question, for example, the big corn companies are marketing their drought-tolerant corn hybrids.
Yes.
But I get the impression that they can't tell you why they're drought-tolerant, only that they've screened so many and found some that perform well.
Yes, and I have to be careful what I'm saying here because I'm being filmed, but I have former students and friends who work in the seed companies who tell me informally that they're not really making headway with drought. So the idea of genomic selection on yield under drought is not, that kind of gets to that phene diagram I showed you. There's so many things that affect yield that it's, take the example of root angle, which I showed you can be good under drought, bad under phosphorus, super good if there's root hairs, it's very complex. If you ignore all that and just look at yield, you'll just get a very noisy signal and you won't know which genes are controlling what. So to me, it seems like you really have to understand the biology at multiple scales, but that concept is not well accepted in the US-based seed companies, in my experience. Because the genomic tools are so powerful and so attractive. You can, $10 to genotype the seed and you get all this information. It seems really great. And it's much harder to think about roots and stuff like that. So, but I believe this is, as the limitations of the genomic fixation are becoming more evident, I think we're going to see a return to an interest in the pheno.
Two questions. One is, have you done anything with endophytes?
We have looked at some endophytes in fescue. We haven't looked at it in maize or bean. Should we?
I suspect probably so, but I don't know. There's not enough data to really predict. The other question is connected to that, and that's one of the other critical environmental parameters is temperature.
Yes.
And there is some evidence that the endophytes affect the temperature response in the plant.
Yes. And then I lead a project in which we have work on high temperature tolerance, but they seem to be focusing on reproductive processes and pollen viability and so on. This could relate to the question that the other gentleman asked about mycorrhizas. It could be a factor that's there in the background, and if it doesn't change the genetic rankings, then we don't see it in our screens. So we're just interested in finding better genotypes compared to the ones we now have. So if they all have endophytes, then maybe we don't have to focus on that. I don't know if that's true. Any other questions?